The Process of DNA Replication by Susie and Anika. Today we will be watching the process of DNA replication in a cell just going from its first growth phase into the synthesis phase, where its DNA will be replicated in preparation for eventual mitosis. Before we jump in, we will be using lots of proteins, so here's a key of how we represent each of them. In addition, we will be using some phrases such as leading and lagging strands and Okazaki fragments. So here you can see that the leading strand follows the helicase when building, meaning it only needs one section of RNA primer, while the lagging strand goes in the opposite direction of helicase and has multiple sections of primer for DNA polymerase to grab onto while building, to try and keep up with the ever-increasing amount of template DNA ready to replicate. These sections are called Okazaki fragments. Here's a quick reminder on the structure of a DNA nucleotide. There are 4 different bases: thymine, adenine, cytosine, and guanine. These bases are also present in RNA nucleotides, with the exception of thymine being replaced with uracil. There are base pairing rules as well, with the double-ringed purines A and G always bonding to the single carbon-ringed pyrimidines T or U and C, respectively. In addition to these bases, there's also a sugar and a phosphate group. Here we see a deoxyribose sugar, which is what differentiates DNA from RNA, which only has a ribose sugar. This sugar has 5 carbons, which is important for bonding with other nucleotides to form the sugar-phosphate backbone. Number the carbons from right to left, and it's important because the number 3 carbon attaches to the phosphate group and the nucleotide behind it, and the number 5 carbon attaches with the phosphate group to the nucleotide above. This creates a sort of right and left system on the DNA strand. This is how we are able to tell which side is the leading and lagging strand, because DNA polymerase is only able to lay down nucleotides in a 5' to 3' direction. If helicase unzips in a 5' to 3' direction, the strand in that direction will be leading, while the strand going 3' to 5' will be lagging. Helicase starts unzipping in the middle of a strand, which is the origin of replication, and creates a replication fork as the base pairs split apart from each other. Next, RNA primase comes in to add a few RNA nucleotides that will give DNA polymerase a starting point to grab onto when it goes to place down new nucleotides. DNA polymerase starts laying down matching base pairs on the template strand in the 5' to 3' direction. In the example shown here, it is in the leading strand, so there is only one primer segment laid down. After that is finished, a second polymerase comes in to replace the RNA with DNA, and then the final protein ligase comes to connect any fragments to each other. The fragments connected here are the primer fragment, which has been replaced by DNA, and the rest of the DNA placed down by the first polymerase. Ligase covalently bonds the sugars together, forming one long backbone. This process is slightly different in the lagging strand. After helicase comes through, primase comes to lay down RNA primer for the polymerase. However, the helicase is moving in a 3' to 5' direction for the lagging strand, and so primase needs to lay down multiple primers as more of the DNA is unzipped so the polymerase can continue laying down nucleotides on the newly unzipped DNA. A polymerase comes in and does just that, going from one primer to the next as the segment is completed, making Okazaki fragments. After this, the second polymerase comes in like normal to replace the RNA nucleotides, and then ligase comes through to connect all the Okazaki fragments. Thank you for watching.